I'm Simon Charnock, Managing Director of Prozomix Limited. We're an enzyme discovery company, 10 years old almost now, and we discover enzymes principally for big pharma companies. We enable synthetic chemists to make new drug molecules using more greener technologies. It's green chemistry. So as an enzyme discovery company, we rely on bioinformatics software to be able to go into our DNA databases, either public databases or more now metagenomics private and proprietary databases, where we've gone into the environment, collected DNA samples and sequenced them for ourselves. But as time goes by, we need to generate more and more enzymes of increased diversity, of guaranteed diversity for the customer. And that meant we needed more bioinformatics, more powerful bioinformatics algorithms. And we're biologists here, not computational scientists. So we actually then attended a networking event run by Innovate UK, where we were put in contact with Newcastle University and the group of Professor Anil Whipat. And they were very keen on working with industry, especially in the northeast of England, where we're both located. And they proposed to generate the software that we required. Unfortunately, that would cost quite a lot of money. And then coinciding with these events was the integrated omics call from Innovate UK. And this actually gave us the financial opportunity, if we were successful in the competition, to have Newcastle University build this cutting-edge bioinformatics software. So we applied along with Newcastle University with a lot of help from Sander Hensig from the KTN. And in the end, we were successful, we were funded, and now that's why the company is able to expand as we're currently doing, because our customers know that the enzyme panels that we're currently developing or have developed recently are really the best in class. And I've spent most of my scientific career in the northeast of England. I went to university at Newcastle University, I lectured at Northumbria University, and that was when we bought our house in Holtwhistle, which is why we started our small biotechnology company, or a little startup, in Holtwhistle. We're not a spin-out company, we're not VC funded. We started very small, close to house, with the idea of establishing all of the biological systems and then maybe moving closer to Newcastle. As it happened, the company's just grown in Holtwhistle, which is a great place to do business. We have a low overhead environment, great local trades, and many people actually appreciate living in a rural, beautiful environment that we have in Northumberland. Along the route, the Nagoya Protocol in 2014 means that we can't just take these DNA sequences from anywhere in the world anymore. We can take them from anywhere in the world, but we have to share benefits back to those territories. And although we'll be very willing to do that as a company, it's a very messy and so far undefined process how you kind of obey the Nagoya Protocol. And moreover, the enzymes for biocatalysis are housekeeping genes, common genes found in most microbial organisms. So we decided to go to the environment of the United Kingdom, to take environmental samples, screen those samples for DNA molecules of interest via a process called metagenomics, using our new software, and then present enzymes to anybody in the world with freedom to operate. FTO is a critical factor when selling enzymes, especially into the pharmaceutical industry. So after a few years of operation of the company, when we'd established some quite powerful gene cloning technologies, we proposed to build what we call our biocatalysis enzyme toolkit. And we offered it via our toolkit concept. And the concept, unlike these other suppliers, is that we would make these enzyme panels and then supply them free of charge to the pharmaceutical industry, where instead of spending $10,000 or $20,000 on a panel of enzymes, they'd be able to screen them effectively free of charge, apart from their staff time. And with just a few milligrams, they can set a whole plate up like this, where they just put 20 microliters in each well, and if any of the wells turns red, then it means that particular enzyme, that native natural biocatalyst that we've evenly sampled from this diversity we've got from the United Kingdom, that enzyme can actually produce the chiral alcohol of interest. And they can find that information out in half an hour just by holding this plate above a sheet of white paper. And basically using innovative products like this, which were developed with Innovate UK money, we've been able to go to new companies and convince them immediately that the enzymes we're offering, the diversity of enzymes, are actually relevant for their processes. And we can generate a global supply of our biocatalysis enzymes right here in Holtwhistle. Obviously, the grant support we've had from Innovate UK, much of that has been supported by the KTN as well, in them offering advice, telling us about the grant calls coming up, maybe giving us some key phrases and angles that we should approach. certainly via the workshops that they put on before the grant call closes. Even to the point of reading through applications and making suggestions of fine tweaks, which they are prepared to do for anybody. If we do our day job well, then we can enable these pharmaceutical companies to develop new products and new drugs, make them cheaper, maybe make drugs that they haven't been able to make before. And next year there will be a new drug launched by a major big pharma company, and we were able to find one of the critical enzymes for that process. And we know here that unless we've done what we've done over the last 10 years, that particular drug would either be delayed or maybe it wouldn't have been launched in the first place.